Okay, in honors biology today, we're going to be drawing mitosis and meiosis. So I'm going to be switching between two sheets of paper. Okay, so drawings of the cell cycle for an animal cell. So we'll be doing our drawings for an animal cell. The same thing basically happens in a plant cell. So mitosis, when a cell undergoes mitosis, it is a type of cell division that produces two identical cells. So that simply means that the cells have the same number and kind of chromosomes that the original cell had. Mitosis will occur in what we call the body cells or somatic cells, except for mature muscle and neurons. Your muscle cells and neurons are no longer going to undergo mitosis to make new ones. The purpose, of mitosis is for growth and repair of damaged or old cells, and the stages in order are PPMAT, prophase, prometaphase, metaphase, metaphase, anaphase, and telophase. Okay, so an interphase, the DNA is in a form called chromatin. This is where synthesis of new DNA occurs. The nuclear membrane and the nucleolus is intact. So I'm going to move a sheet of paper over. I'm going to draw my cell. This is your nuclear membrane. This is your nucleolus. Out in the cytoplasm, you'll have two structures called the centrioles. We'll talk about those in a second. And the DNA, and I'm going to do it in red and blue to indicate for mother and father. Obviously, in your cells, it's not color-coded. So we're going to label this chromatin, which is your DNA along O-M-A-T-I-N, which is your DNA along with the associated proteins, okay? Now, this cell, so we're going to assume this cell is going to undergo interphase. And so in interphase, the chromatin will coil up around your proteins and appear as two sister chromatids held together by a centromere. And then the nucleolus will disappear, and then the centrosome has duplicated, and each one will move to opposite poles to start making spindle fibers. Okay, so centrioles, centrosomes, basically the same thing. Okay, so we're going to assume this cell undergoes mitosis. So this was interphase. And you're going to be drawing your drawings here, linear, okay? So it's going to enter into what we say is, so it's going into mitosis. So it's going to enter into prophase. So this is going to be prophase, so it's the same cell. Okay, so your DNA, so your nuclear membrane is still intact. The nucleolus has disappeared. Your centrioles or centrosomes have moved to opposite poles. So we're going to end up here and here. And they start to make spindle fibers, which are made up of microtubules. So these are your centrosome. And then the little dotted lines are the spindle fibers. Okay. Now the DNA has condensed, and so now it's going to be in rod-like structures that you can see. So this is going to be a chromosome from Dad with its copy attached. And here's going to be a little one from Dad with this copy attached. And then let's do a squiggly one. I hope these show up for you. And then Mother has some identical to it, same length. Okay, so when we look at this cell, let's talk about a couple of things, or this nucleus. This is a pair, this is a pair, and this is a pair. So if you take one from each pair, so let's just say one of each of the blue ones, that's a set. And then if you take one of each of the red ones, that's a set. So this cell has 2 sets, so it's 2 in, 2 sets are diploid, and its chromosome number is 1, 2, 3, 4, 5, 6. So if we were drawing your body cell, we'd have to put 23 pair in here. So we're going to stick with 2 and a 6. Now let's talk about this real quick. Whenever this is a sister chromatid, and this is its exact copy, okay? And they're connected by something called a centromere. So we have 2 sister chromatids. Attached by centromere. So as long as they're attached, we call them sister chromatids. Once they separate from each other, then we'll call them chromosome. Okay? Okay, your next phase is called prometaphase. And the main thing that happens here is the nuclear envelope will break apart. The microtubules will move into the nuclear area and attach to the kinetochore on the centromere of the sister chromatid. So the kinetochore reminds me kind of like a hook, like a hook and an eyelet on a shirt. It's like a hook, and it's going to allow the spindle fibers, to attach to it. Okay, so now we're going to go into prometaphase. So now the cell looks like it did earlier, shouldn't be misshapen, and our nuclear membrane is breaking apart, okay? And your spindle fibers will get longer, and they come in, and they actually attach to a chromosome, to the centromere, okay? So let's go back in and draw our chromosomes in. Okay. Okay, the next phase is metaphase. This is metaphase of mitosis. The microtubules will move the sister chromatids to the equator single file. Okay, so you draw you another cell. So this is metaphase. And you're just going to put these on the equator single file. They do not have to be next to each other. So I'm actually going to separate them out. So I'm going to put the big one from Dad here, and then I'm going to put the little one from Dad here, and the squiggly one from Dad here. And then I'm going to put the big one from Mom here, the little one from Mom here, and the squiggly one here. So that's what we mean single file, but they don't have to be next to each other. They still have their centromere holding them together. I don't often show it because I get kind of lazy. Don't tell anybody. Okay, so it was the job of the spindle fibers to move those sister chromatids around. So they're kind of like the pulley system of the cell. So these dotted lines, once again, are the spindle fibers. Okay. Okay, next phase is anaphase. So in anaphase, the centromere that holds the sister chromatids will replicate itself. And so the centromeres will separate, allowing sister chromatids to come apart. Now we call each of them a chromosome. The kinetochore microtubules or spindle fibers, whatever it is you want to call them, will drag the chromosomes to opposite poles. So now, I'm going to have to come around this way. I'm running out of room. So now we're in anaphase. And so basically everything from this line across will go to this pole, and everything down will go to this pole. So now you're going to have these being dragged to the opposite poles. So you're separating them from their copy. Here's the big one from Dad. Here's its copy. Here's a little one from Dad. Here's its copy. Here's a squiggly one from Dad and its copy. And then the same for Mom. Here's the big one from Mom. Here's a little one from Mom. Here's a squiggly one from Mom. Okay. And once again, they are being dragged to the poles by the spindle fibers, kinetochore microtubules, whatever. Okay. But now you would call them each a chromosome. Okay. And then our next phase is telophase. At some point during telophase, the nuclear envelope will reappear, the nucleolus will reappear, the chromosomes will start to uncoil again, and they'll go back into the form of chromatin. And then during telophase, at some point, cytokinesis will start to occur, and that is where the cytoplasm will pinch in at the center of the cell, and it forms something called a cleavage furrow, and that pinches the cell in two. So we'll end up with two cells. So we're going to end up drawing two different drawings here probably, okay? So now our next phase is we're going to draw, well, let's do intermediate telophase. So this is cytokinesis is beginning to occur. We're going to go ahead and put our nuclear membrane back in, put our nucleolus back in, okay? And now what I'm going to do is I'm going to separate, I'm going to take my chromosomes and stretch them out so you can see them again. Okay? This is cytokinesis, so you call this furrowing or cleavage furrow. And it's eventually going to go all the way across, so you're going to end up with two different cells. Here's your nuclear membrane, here's your nucleolus. Now, when you go to count, you still have one of each of the blue ones, so that's one set. You have one of each of the red ones, that's a set. So this cell has two sets. It still has 6, 1, 2, 3, 4, 5, 6. This cell has two sets with 6. So we started out with a cell with 2 N of 6. You make two new cells that are also 2 N of 6. And then each of these cells will go into, they become individual cells, and they go back into interphase. So the DNA just starts to get all squiggly again. Okay, and they hang out in interphase until they reproduce to make two new cells. Okay, so that is it for mitosis. Now, the next thing up on your notes, oh, let me say this first, excuse me. Plant cells, so plant cells will do the same thing. The cells will be rectangular instead, okay? They don't have centrioles, but they will still form spindle fibers or microtubules, okay? They do not have furrowing to occur during cytokinesis, but instead a cell plate will be put down. So whenever they are finished at the end, so here's your original cell. It has one nucleus with the DNA in it. It's going to make two new cells, each with their own DNA, and then this is the cell plate. Okay. All right. Now, if you'll notice, the next thing in your notes says regulation of cell cycle, and then I've put words like external factors, internal factors, apoptosis, etc. Okay, so what I want you to do on this section is you're going to make up your own notes. You can use this sheet and the next sheet as a guideline, or you can come up with your own, but you must include all these terms. And then these notes will go along with the study guide that's titled Regulation of Cell Cycle, Asexual Reproduction, and Multicellular Cells. So you're going to read that section in your book, and then let's see if I can tell you the section number here in a second. Yeah, it's going to be pages 140 to 151, and then you're going to send me a copy of your notes that you generated, okay? Now, we're going to move on to meiosis. So meiosis is another type of cell division, but it is called reduction division. So it's called reduction division because during the process, you're going to reduce the number of sets from 2 sets to 1 set, and you're going to cut your chromosome number in half, okay? So you'll end up also making up to 4 cells, depending on who we're talking about, male or female. So it is a cell division that produces gametes. So gametes are like egg and sperm, and they will not be identical. In males, this process is called spermatogenesis, spermatogenesis. And in spermatogenesis, for each cell that undergoes meiosis, a male will produce 4 sperm. In females, it is called oogenesis or oogenesis, oogenesis. Each cell will result in only one viable ovum or egg, because what happens is when they divide, the first round, the one cell doesn't get as much cytoplasm. It's called a polar body, and it will atrophy. It won't carry on. And so for meiosis 2, you only have one cell going into meiosis 2, and then that cell, the cytoplasm, once again divides unevenly, makes another polar body, so the end result is that a female will only produce one ovum or one egg. At least we would have multiple children every time. Okay, it will occur in what we call the gonads. So in a male, that is the testes, and in a female, that is the ovaries. The purpose is for several things. Number one, it produces what we call gametes or sex cells, sperm and egg. It will reduce the chromosome number by 1/2 to maintain the species number, and it causes genetically different sex cells. Okay, stages of meiosis. So we're going to have meiosis 1, and the purpose of meiosis 1 is it's going to separate homologous chromosomes. So once again, I'm going to be going between several sheets of paper, so you'll just have to forgive me, and we're going to have quite a few more drawings. So I left enough room, hopefully on the side, for you to do your drawings. Okay, so this cell went through interphase and replicated its DNA in the S phase, okay? So we're assuming that you already realize that. So prophase 1, homologous sister chromatids pair up, and so there should be a pretty good picture of that on page 167 of your textbook. And they form something called tetrads. Tetra means 4. During prophase 1, synapsis will occur. So synapsis is where these homologous chromosomes will pair up precisely gene to gene down their entire length. And when they do that, something called crossing over occurs. And so that's where parts of DNA are exchanged between homologs, or between mom's chromosome and dad's chromosome. And the chiasma is the site of crossing over. Now, I'm not going to show this, but I'll show you where we're going to have crossing over occur on a couple, just so you can see what we're talking about. The centrosomes will move to opposite poles once again, just like they did in mitosis, and form the spindle fibers, which are made of microtubules. Okay, so I'm going to have to draw a little bit more organized since we're going to have more drawings. Okay, so we're going to have prophase 1. So this is prophase 1. I'm going to just title it, I'll just call it prophase 1, I guess. Okay, so we have our nuclear membrane. Okay, and you probably ought to draw it breaking up. I didn't, and I'm not going to put the nucleolus in it. Okay, so we're going to do the same chromosome number we did earlier. And so red is going to represent mom's, blue is going to represent dad's. Get going here in a second. There we go. Okay, so here is dad's chromosome, and here is mom's chromosome, and they're real close together, and they get so close that some of their arms like touch and they exchange DNA. So I'm going to put a little bit of mom's on dad's right here, and I'm going to put a little bit of dad's on mom's. Now here's the deal. Don't go crazy doing crossing over, because you're going to have to do it every stage you go through. You carry it with you. And then here's one from dad. I'm going to do a tiny little one from dad, a tiny little one from mom. Okay, and then I'm going to do a squiggly one from mom and a squiggly one from dad. And let's do some crossing over, okay? So a tetrad is, you see 4 arms, 1, 2, 3, 4. So that's a tetrad, a tetrad, a tetrad. Okay, once again, this cell has 2 sets, so it's diploid 2N, and has a chromosome number of 6. Okay, so that is prophase 1. Okay, the next phase is called metaphase 1. In metaphase 1, homologous chromosomes pair up at the equator. So if you go back and look at your mitosis drawings, they are single file. Here they will pair up. Okay. So I'm just going to draw it next door. So this is going to be metaphase 1. And you should have your centrosome, and your spindle fibers forming. Okay, so my mom and dad's chromosomes pair up at the equator. Oh shoot, right here, I'll do this. Okay, and you got to take your crossing over. Make this a little bit longer. Okay. And remember, it is the job of the spindle fibers, microtubules, to position them. But the thing is, they are paired up at the equator. I probably should have had this to pair up a little bit higher. Okay. Okay, the next phase, anaphase 1. Paired homologous chromosomes separate and are dragged to opposite poles by the spindle fibers. So what we're saying is now this chromosome with this copy, this one with this copy, and this one with this copy are dragged to that pole, and then this one, and this one are dragged to that pole. So this is going to be, I forgot my 1 here, goodness, anaphase 1. Okay, so now you're going to have like X's being dragged to the pole. Don't forget you got to have your crossing over with you. Gets to be a pain. And they were dragged to the pole by the spindle fibers. Okay? And then the next phase is telophase 1. In telophase 1, the spindle fibers will disassemble. Cytokinesis will occur, so it's going to divide, so you're going to end up with 2 new cells. However, each cell will have has only 1 set, but they have to go back through meiosis 2 to separate them from their copy. Okay? So here's going to be telophase 2. I'm going to draw the cell a little bit bigger just so I can get everything. So cytokinesis is going to start occurring. Okay? Your nuclear membrane will reappear. It's going to divide and make 2 cells. I'm going to stretch it out so we can see what's going on in here. So this cell has a squiggly one from Dad, and then down here we have a big one from Dad and a little one from Dad. And then we have a big one from Mom here, and a little one, and a squiggly one from Mom. And we've got to take our crossing over. So let's put, oops, okay. And then get myself confused. Okay, got it. So we have 2 new cells, and they each have only 3 chromosomes in, but here's the deal. They've got their copy attached. So we're going to have to take them back through meiosis 2 and see what happens. Okay, so meiosis 2, the job of meiosis 2 is to separate sister chromatids. Now, prophase 2 looks just like telophase 1. So I'm not going to redraw it. I'm just going to draw it. I'm going to write that name underneath it, okay? So I'm just going to say telophase 1, prophase 2. And so what we're going to do is we're going to go back in this direction. Okay. In metaphase 2, the sister chromatids are at the equator. Okay, so now the thing is you're going to have 2 cells, 2 distinct cells. This is in one cell, this is in the other. So put those chromosomes at the equator. And remember, once again, even though I don't always show it, it is the spindle fibers and microtubules that place them, move them around. They're like the pulley system. Okay? So this is metaphase 2. Anaphase 2, the sister chromatids will separate. Now you can call the structures chromosomes, and they're going to be drugged to the opposite poles by the spindle fibers. Okay? So once again, you have 2 separate cells. This is going to be anaphase 2. Okay. You're going to separate them from their copy. So this one goes to this pole, this one goes to this pole. And once again, you got to take your crossing over with you. I'm going to make this one a little bit longer. Okay. Now, in telophase 2, the nuclear membrane and nucleolus will reappear. Each cell now has only one set of chromosomes, and they are going to be genetically different. And then cytokinesis will occur, and it will take each of those 2 cells and divide them in half, and now you have 4. Okay? Okay. So this cell here is going to divide, this cell here is going to divide. You can put your nuclear membrane back in. Put your nucleolus back in if you want. Should have made them a little bit bigger. So this one's going to have a big red one, little red one, little red one. This one's going to have a big blue one, little blue one, squiggly red. And got to put our crossing over in if we can find it. Here we go. We'll just put it in there someplace. Okay. Goodness, glad we don't have to direct this in our cells. Okay, so at the end of meiosis 2, at the end of telophase 2, which is the end of meiosis 2, you end up with 4 cells. Now, instead of them having 6 chromosomes, they each have 3, and they each have 1 set. So we say they are each N of 3. N of 3. So you have cut your set number from 2N to N, and you've cut your chromosome number in half. Okay? Now, one reason meiosis needs to occur is to maintain the species number. So if, let's just say this is a sperm, and here's an egg. Okay, so here's a sperm, here's an egg. He would have 3 chromosomes, and she would have 3 chromosomes. And when these 2 unite through fertilization, then you're going to get a cell that's going to return back to that original number of a chromosome number of 6. Okay, 2 sets. So this would be N of 3, N of 3. They get together, and that makes a cell with a 2N of 6. All right, I hope you could see that. If not, there are many good videos. You can go on the internet and type in animation of mitosis, animation of meiosis, and you can see this happening in probably a lot better drawings. Okay, so now this will finish up your information. So now you can do the study guide that is titled Mitosis and Meiosis. Thank you.